Hi, welcome to MedBits, your exam savior. Today, let's discuss another important topic in physiology, oxygen dissociation curve. Again, this is a short question for the exam. Okay, let's start it. The oxygen hemoglobin dissociation curve, also called the oxyhemoglobin dissociation curve or simply oxygen dissociation curve, is a curve that plots the proportion of hemoglobin in its saturated, that is oxygen laden form on the vertical axis against the prevailing oxygen tension on the horizontal axis. This curve is an important tool for understanding how our blood carries and releases oxygen. Specifically, the ODC relates oxygen saturation SO2 and partial pressure of oxygen in the blood, which is known as PO2, and is determined by what is called hemoglobin affinity for oxygen. That is the most important point you need to write when you are writing about the ODC curve. Hemoglobin affinity for oxygen is how readily hemoglobin acquires and releases oxygen molecules into fluid that surrounds it. Now let's look into the oxygen dissociation curve details once again. Don't forget to draw the diagram. It's very simple. The x-axis is partial pressure of oxygen PO2 and y-axis is the oxygen saturation SO2. As seen in the image, oxygen dissociation curve is sigmoid in shape, which has high physiological significance. Hemoglobin in the blood is the primary vehicle for transporting oxygen from lungs to tissues and organs. Also for carbon dioxide back to lungs from tissues and organs. Hemoglobin's affinity for oxygen increases as more molecules of oxygen bind. This results in increase of oxygen partial pressure until the maximum amount that can be bound is reached. As this result is approached, very little additional binding occurs and the curve levels out like a plateau, that is the upper portion of the curve, as the hemoglobin becomes saturated with oxygen. In the lungs, at the alveolar capillary interface, the partial pressure of oxygen is typically high. That means the plateau shape is happening in the lungs, and therefore the oxygen binds readily to hemoglobin that is present. As the blood circulates to other body tissue in which the partial pressure of oxygen is less, the hemoglobin releases the oxygen into the tissue because the hemoglobin cannot maintain its full bound capacity of oxygen in the presence of lower oxygen partial pressure. This is happening on the steep curvy areas of oxygen dissociation curve. You can look into the diagram once again so that this said matters will be more clear to you. Okay, now let's look into this oxygen dissociation curve once again. This is just an extension of the previous slide. This is the plateau portion and this is the steep portion. This plateau portion is happening on which part? As we said in the previous slide, that is happening in the lungs. See, here is where we can say that the hemoglobin affinity for oxygen is high. Even for very high partial pressure, the hemoglobin oxygen saturation is almost constant. And on this steep area, this is the portion when the oxygen is being given out for the tissues, that is happening in the tissue level. Now just look into the partial pressure at 60. The pressure above 60 mmHg, the standard dissociation curve is almost flat. What that means? That means the oxygen content of the blood does not change significantly even with a large increase in the partial pressure. See, from 60 mmHg, the partial pressure has increased to 100. But still, this oxygen saturation, SO2, is almost the same. It's a plateau. Here we discuss about how the oxygen dissociation graph can have a right shift or a left shift, and also about P50. Okay, there are some factors for both the shifts and it is detailed here. Either you can just read it out or we are explaining it in the next slide. Now it is about P50. P50 is asked in a 3-marker sometimes. Okay, and these 3 points you need to write when you're writing about the oxygen dissociation curve. So let's look into that. As I had shown you in the last slide, P50 is the partial pressure of oxygen in the blood at which the hemoglobin is 50% saturated, typically about 26.6 mmHg for a healthy person. This is known as P50. It is a conventional measure of hemoglobin affinity for oxygen. In the presence of disease or other conditions that change the hemoglobin's oxygen affinity and consequently shift the curve to right or to left, the P50 changes accordingly. Now how the right shift is happening. Let's look into that. When the curve is shifted right, what happens to P50? Increased P50. That indicates a rightward shift of the standard curve, which means that a larger partial pressure is necessary to maintain a 50% oxygen saturation. This indicates a decreased affinity. The curve is shifted to right, that is lower saturation for a given PO2 by higher partial pressure of carbon dioxide. Greater acidity means lower pH and higher temperature. These are the factors affecting a right shift. Now about the left shift. Lower P50 indicates a leftward shift, which means lesser partial pressure is only necessary to maintain a 50% oxygen saturation. This indicates a higher affinity. Congratulations, we have come to end of oxygen dissociation curve, and make sure that you are drawing the diagram and writing about its vertical axis, horizontal axis, the importance of oxygen dissociation curve, the hemoglobin affinity towards oxygen, the terms like P50, its value, why the oxygen dissociation curve should shift right or to left. Okay then, good luck guys. Bye. See you again with another topic.